Yeah, thank you very much. Yeah, my talk is gonna be titled Microscopy with a Tilt. So it's a bit of foreshadowing. So what we're gonna do is we are looking at image planes that look a bit different. But first off, I'm joining from Norway, very far up in the world. So if you see my mouse, it's all the way up here, about 10 hours north of the polar circle by car. And this is how it looks pretty much right now. Bit less sunlight. We are in the Arctic night. So a lot of snow, good for skiing. And what we like to do in Tromsø a lot is fluorescence imaging, both outside, but also in the lab. And our prime method we are using, that's really anything. We have confocals, we have light sheets, we have super resolution. What I'm going to talk about is a very special kind of method that could be used for any of these. And that's rolling shutter line scanning. So what's that? In the easiest case, if you have a microscope with just one objective that's looking at one line, so you can see here, if you look from the top onto a sample, that line is illuminated and you scan that through your sample. So what you end up with on the camera is you're scanning one line through your sample, and that's going to be illuminated. So it's a very fast way of doing confocal microscopy. Well, not confocal, it's with a line that's confocal. And usually what you do is you illuminate this is the back focal plane, you illuminate the whole back focal plane with a line and then get a nice diffraction limited line in your sample. That's one way of doing line scanning in a confocal method. But that's not the only one. What you could also do is you can build a light sheet microscope, and instead of having a light sheet, you project a thin a pencil beam, and you scan that sideways. If you look from the top, and you put that on the camera, and you scan that over. And that's also one way of getting rid of a bit of out of focus light there. And you can scan another way, and that is Reto Fjölker's brainchild a lot. And that's axially scanned light sheet microscopy. So there you're basically sweeping the waste of a highly focused light sheet into the direction of the illumination objective, and you conjugate that with the camera. In this case, you're actually having kind of a very thin sheet made. But there's a fourth way of doing it. We don't need to have light coming out of one objective straight or of another objective straight. We could just come from the side. And that actually gives a lot of cool things that we can exploit, specifically for two types of microscopes that I'm going to talk about today. One is a technique that we published a few years ago, scanned oblique light sheet instance sectioning, so SOLIS for short. And the other one is oblique plane microscopy. That's a proper light sheet technique already published in 2008, but really started off very recently with the introduction of the so-called snouty objective. We'll come to that. So let's start with number one. What is this? Well, before we go there, it's composed of two parts. It has light sheet illumination at an oblique angle, and it combines it with multi-focus imaging. And that gives a way of doing light sheet microscopy from a single objective. So let's start. What is line scanning in detail if you look at all the nitty-gritty details? You basically have a scanning system, and when you scan that galvo mirror, a few lenses are missing, that's okay. You see that this scans around, left to right, and on the camera you can see that it's also scanning left and right. Now this is on the camera. These are out of focus planes, right? So you can see that we only get the light that's produced here that's confocal onto our camera. So it's focused there. This is another view. So if we look from the side, then this cross section here, that is actually an entire line. So, right on the camera, this would be the line that we are scanning. Now, when we are scanning, we produce an image. very naively, this would, of course, just be camera is exposing with a global shutter. You get all of the light that's produced in the sample onto your camera. And if you do that, and I did that for you, then you get an image like this. So these are mitochondria in human cardiac tissue. So it's heavily scattering, and you can see there's a lot of out of focus light, but there are also a few mitochondria in there. But ideally, we want to get rid of out of focus light as we do in confocal microscopy. Now with this setup, what's nice is you can see here, what I draw you in here is that all the light that is generated in different planes that are not the image plane, that's kind of ending up in our image plane, our nominal image plane. Somewhere like here. So we have light that is generated somewhere above the focus plane and somewhere below the focus plane. All that ends up where our nominal image plane should be, really sharp. Now, if you scan that, it's clear that all of that out of focus light ends up on the camera chip as well. And to get rid of that, what we can do is we make use of a rolling shutter. Basically, only these pixels around this very tightly focused beam that we are imaging from sample space onto camera space, only that one is really in focus, and all the out of focus light is actually spread out a lot. So this is a regular confocal idea. Something that's outside of your confocal pinhole largely gets blocked by that pinhole. Just in our case, the pinhole is replaced by a digital slit that rolls along with our illumination line. But there's a better way of doing that, even just in good old line scan confocal. And that is by using oblique line scanning. By coming with an oblique angle, you're basically generating out of focus light such that it ends up next to our line. So all this out of focus light that we would be generating here is actually ending up on the camera next to our illumination line. So now when we use our rolling shutter, we block a lot more of that light. And now if we scan that, we get a really nice and crisp image. So it's the same one. Now we just use the rolling shutter with the correct settings and an oblique light sheet. Now we get that image. And you can really see what a big difference that makes. And it's a super simple microscope to build. Just line scanning though, right? So how good really is it? We can look at that from a theoretical perspective. So what we are basically doing is these are optical transfer functions for both cases. Like this is just on axis and this is with the oblique light sheet. In both cases, let's say we have the same numerical aperture for the detection objective and also for the illumination, well, line. When we calculate what the effective support is of this optical transfer function, we basically convolve these two together. And then our readout is, well, what is the axial extent here? The fattest one, that's the axial resolution for one definition. And the degree of optical sectioning is how good you're providing support around this 0 kx, ky point. So along the kz direction, how much is that? In wide field, that's very close to 0. Now, if we do this convolution, basically what we do is we just smear it out over each other, right? That's what a convolution is. And then we can look at what is the support that we end up with in both cases. And what you can see is that actually the support, both for optical sectioning, so on the 0 crossing, as well as the axial resolution, so the maximum extent, that is both higher if you just use an oblique light sheet. So if you want to build a line scanning confocal system, just take your illumination part and just nudge it to the side by a few millimeters. So you're illuminating with an oblique line and you almost double your optical sectioning and resolution. It's a very simple trick to get a lot better image quality. You can, of course, do that also a bit more rigorous with proper 3D simulations. So if this is a 3D image of beads, a 3D volume, and we simulate the same thing, then this would be the on-axis illumination for line scanning. This is the respective OTF cut through in the middle. And this would be the oblique illumination. So you can see that you get a lot more support and a lot better optical sectioning, basically for free. This was the first part of SOLIS. So let's look at what is the second part, this multifocus optical imaging. So multifocus optic is, well, what the name kind of suggests. You look at something, so if you look out of the window, and I did that here, had a lens here and put it on a camera, this is the image that you get of this lamppost. And if we move the camera a tiny bit to the front, so we are out of the focus of this lens, then we're looking behind, we're focusing behind that lamppost. And if we do it the other way, then we're looking in front of the lamppost, and it kind of gets blurred out. And what multifocus imaging is, basically generating these three images at the same time onto one camera chip. So really we want them next to each other and all sort of focused on different parts of a camera sensor. And this is actually fairly easy to do. All you need to do is put a beam splitter cascade in. So these are beam splitters. One portion of the light splits off, one goes through, let's say a 70-30, 50-50, and this is just a right angle mirror. And based on what type of beam splitters you use here, you're changing the optical path length to all of these. And the optical path length change that actually focuses on different parts in your sample. Generally, that causes a bit of aberrations, but within limits, you are still getting close to diffraction limited performance. So now we have a way of getting an entire image volume put onto a single camera chip, just placed next to each other. So let's add that to our line scanning system. What happens now? Well, all of these image planes that we had here, the nominal focus one, but also the two new ones that we are generated with this multifocus element, they're basically ending up next to each other. So you have one line going through a sample, one sheet of illumination, and each time you cross through one of these image planes, you're putting a line down on your camera next to each other. So basically, you have an image plane linearized onto a line. So it's a type of light sheet imaging system, just that every single depth in your sample becomes its own part on the camera chip next to each other. In the light sheet, you would get all of these kind of stacked upwards. And with SOLIS multifocus imaging, you get them sideways. But now the cool thing is, when you scan through the image, you scan the entire volume. What I drew here is the autofocus light though, and that's also quite important to take care of. You can see that all of the autofocus light that's generated by these, it all blurs over to all of the other image planes. But nicely, as it was with our single line oblique light sheet, oblique confocal, all of that autofocus light ends up pretty much outside of where we want to image. So now if we take our rolling shutter, and because all of these image planes are exactly next to each other, we can rolling shutter confocal all of these different lines. So it's basically light sheet imaging, but confocal, right? So it's plane scanning confocal microscopy if you want. And by scanning that plane, you end up with an entire volume with, well, truth be told, a very thin volume, just 3 planes, but you can get the principle, right? If you use a different multifocus element, that scales. You get these 3 image planes and you just stack them up and you get your volume. If you have a sample that is not too dense, but a bit sparse, you might be very well off just interpolating between these different planes and you get a nice image. And it's remarkably simple to build as well. So this is a very simple way of building an oblique line scanning system. As I showed before, you basically you take a laser, fiber coupled, collimated. This one is a cylindrical lens. So you're basically generating somewhere in this space your light sheet. This is a beam splitter. go through a scan lens, put a scanning mirror here, go through the same lens, super simple to align as well, because it's all just 4Fing. Go this way through the scanner. Here's another tube lens that goes into the back focal plane, and you generate your light sheet here. If you want this to be oblique scanning, just knock it a bit to the side. So this lens here, like this entire part here, you just move that a bit to the side. And you have your oblique light sheet scanning system. All the rest, this is just a normal microscope. Make sure the camera is in the right orientation, so the scanning direction matches with the rolling shutter direction, and you're good to go. And for Solus, all you do is you just add this beam splitter cascade in here, and you can image volumes. That's it. That's just 2 beam splitters and a right angle prism. Here's a 3D drawing of it, so it's really compact, and it actually fits into, I think, what's that? It's like 50 by 70 centimeters. Build a box around, doesn't take up that much space, and you can do 3D imaging. And this is just on a tiny little breadboard. This is this entire system I have here. This is a collimator, there's a lens, here's the beam splitter, here's your objective or scan lens that goes on the galvo. You go back in, and up you go through dichroic into your sample and go down, and a bit behind this thing, there are the beam splitter cascades that go to camera. And this is how it looks in practice. Every time this dithers, we are basically collecting a volume of 3 planes, and this is the beam splitter cascade that you can see in here, where the fluorescence goes, and then it goes off to the side to the camera. The spacing between these planes, importantly, that's a bit dependent on the optical elements and the magnification that your overall system has. So in this case here, I used a 40x objective, and that makes with the beam splitters I'm using for a 12 micrometer spacing. So it's quite a lot. And this is just a very thin BPAE commercial sample. And as you can see, without this rolling shutter, we're basically just having a multi-focus imaging system. And you can see all the autofocus light. If we match the rolling shutter to our scan, then we get rid of that autofocus light. And that works with 12 microns, it works with a different objective as well. And you also get a tiny bit of resolution improvement. In principle, in one direction where you're tilting your oblique light sheet to. I don't see really that much improvement in practice. It's a few nanometers more with a high NA objective, but it's really the optical sectioning that's the main point. This is now using a different objective, 100x, 1.35 NA silicon immersion. Different cells, I believe. And you can see that the scale was 5 micron. So we get a tiny split between the image planes. So these are now, well, our 3 different image planes. I was always showing what the camera sees, but if you scan through the sample, you can actually see that there's an image generated somewhere else along the Z axis. And all the autofocus light from these is gone. So that was very thin stuff without 3D structure. But actually what we want to use this for is for actual 3D stuff. And that's a project I had together with Åsa Birkesdottir, who works in the hospital, university hospital, pretty much directly next to our physics lab. And she's a clinician working mostly with cardiac tissue and researching the heart. And I can tell you, researching live heart stuff is difficult in humans because no one wants to donate it. So we need something else. And a very good solution for that is you just pay someone to give you red cells that make for cardiomyoblasts. It's a very simple solution, but it's very far removed from heart muscle. You can take skin cells from humans and reprogram them into pluripotent stem cells and differentiate them into cardiomyoblasts. So you can get this twitching heap of cells. So that's already a lot closer. But really what you want is you want to cast them onto special racks like these. And then at some point, you will actually generate proper heart muscle. And this is heart muscle tissue that's very close to adult cardiac muscle, like it shows all the hallmarks that you would have. From an imaging perspective, also, it's quite dense, it's beating, it's big. It is very hard to get to because it's on these racks. So a normal light microscope with like orthogonal objectives either lacks the resolution to see individual mitochondria, which is what we're interested in, or you just can't get to it because of steric hindrance. So you need a single objective solution. And to get rid of autofocus light, ideally you need also something that can remove autofocus light instead of just not generating it, because of the scattering, you would get it anyways. So regular light imaging is quite challenged by this sample. So yeah, as I said, a lot of challenges, but perfect for this SOLIS method because we can tackle all of these in essence. So if you take that sample and put it on a microscope, this is what you see in wide field, and that's actually what I showed in the beginning, right? That's a lot of autofocus light in wide field. If you do regular multi-focus imaging without the rolling shutter, then this is the different planes we get. So this is illumination, and then using our beams with a cascade to get these different images of different focal planes, about 1.5 micrometers apart. And that gives you this image. So you can sort of see the mitochondria, but it's nowhere near what you get if you do this rolling shutter imaging, where you get every single image plane optically sectioned. And when you then combine that, you can really see where are all the mitochondria in the sample. This is very close to the cover slip, actually. So the image quality is diffraction limited. And we can see really the mitochondria in different image planes very clearly. Now, this is just color-coded maximum intensity projection, but it actually works in 3D as well. So now we do the same thing and we scan just through the sample for a few microns, and about 25 micrometers into the sample, you can still get the resolution of individual mitochondria. After that, it kind of fizzles out. Now, what I'm showing here is just one of these different planes. This would be just the oblique oblique line scanning system. If you do the multifocus imaging, then every one of these images that I showed you is actually then a volume that spans about 2 to 3 micrometers. So you could just use that to pick the well cell. Usually these cells turn out to be quite flat. Pick out the cell that you're interested in and follow it in 3D. Every single image is one volume. Some technical details. You can use the same method to do super resolution. We wanted to do that using single molecule localization. I'm not going to go into too much detail how this works. There's a nice visualization from Ricardo Enriquez. Basically, you make fluorophores blink and you locate the centers of these sparse blinks, and that kind of gives you a higher resolution image when you sum up all the localizations. And of course, you could do that with SOLIS as well. The problem is this line scanning is quite fast, and usually what you want is you want to get all the photons from one on cycle. But there is a new or fairly new type of imaging mode, which is called programmable scan mode or light sheet mode, or something along those lines. Most microscopy companies have a different name for it. It's all the same. Basically, it allows you to change the width of this rolling shutter that's going through a sample, and also the speed at which it propagates. So if you want to move slower through your sample and get all the photons within one blink cycle, you can do that with this type of camera. You couldn't do it with a old school, very cheap CMOS camera, because that is always running at the same speed. And basically what it does is it just adds a delay. It just goes to a new line and then it just waits until it propagates further. So within one line time, and it's usually around 10 microseconds, you can scale that up by n times whatever this line time is. And that's how you can have a longer dwell time effectively. And then you can do super resolution imaging. Now we wanted to do that, but then Drauschi and Mine published this nice paper where they used exactly the system as well. So it's the same style as Solis. It's an oblique line scanning system. And he used that together with single molecule localization. And he showed that, yeah, this actually works nicely. And you don't need anything special. It's just in the camera if you have the right camera, and it works out of the box. Another thing, technical detail, if you want to do thicker volumes or more image planes, then how would you do that? So very generally, this is the formula how to get the spacing in a beam splitter cascade. So the spacing between these image planes is the length that the light goes through one of these. So let's say this is a beam splitter cube with a, I don't know, half-inch size. Then this would be 12.7 millimeters times the refractive index of your sample, so 1.3, 1.4 usually, and you divide that by the magnification squared. So with a 100x objective, that's a lot, and the refractive index of these elements, so 1.5 roughly. So with these numbers I gave you, 12.5, 1.3, 1.5 here, and 100x, you get about 1 micron plane separation. If you want to change that, you change the magnification of the objective. That's the easiest to change it. Or you change the beam splitters themselves. But again, this just is 3. But there are different ways of doing multifocus imaging. There are a lot of publications on that. So you could just use a compound element where you don't get linear splitting off in a cascade, but it just splits off multiple parts. So there you can get up to 8 planes at the same time with a much narrower spacing, in fact. That's defined by this step here. Or alternatively, you get a multifocus grating. So that's a grating that you put in a pupil plane, and it's a grating that is warped. That means every single diffraction order that you get puts a different defocus term on that diffraction order. And based on the grating period that you're choosing there, it's kind of scaled, but the general average grating period, that determines how many of these planes you can fit into an image plane. We have most of these in the lab. So we have our beam splitter cascade. We actually made one of these, a set splitter that splits multiple image planes up to 9 in our system. And we started working with these multi-focus gratings as well and make them in our lab. We don't have a clean room yet, so this is the best we have so far. A really good master student who got that running, and that actually resulted in the paper. I'm not going to go into the detail how this multi-focus grating really works in practice. There is a QR code if you want to go to that YouTube video. 3.5 minutes you can spare to learn about that. But in very short, you have this grating that is kind of has a grating function. This balances the intensities in all of these split image planes. And the multi-focus grating is used to generate the required defocus in all of these. And then you can generate a stack. And this has been shown by Sarah Abramson to go all the way to about 25 planes. So that's actually a lot. Now the trick is, this is not a single camera, but she had someone stack a lot of cameras together and actually build 5x5 cameras into an array. Well, we don't have that, but that's okay. We have one camera here, and we just tried out our home-built multi-focus grating in reflection mode. And it turns out you can actually make them without a clean room. So now we are scanning just I think a 4x objective or so. So the plane separation is hundreds of micrometers, and it's not even fluorescence. But it kind of works. As you go between these image planes that are recorded, this is just a blending through between them. You can see that we go into focus and we go out of focus. And this is the paper how we produce these with just a direct laser writing system. That was also home built. So yeah, that's how you scale them up. Now, what I have to tell you though is scaling up means every single one of these cameras sees less and less light. Because in effect, you're splitting up the light. Here, every single image plane is a third. Here, it's 1/25th. So it's gonna be a very dim image at some point. If you want to image something a lot bigger, with a lot less intrinsic fluorescence, you might want to look at a different method. And that leads me to my second, I think I still have some minutes left, my second microscope I want to talk about, and that's oblique plane microscopy. There, the idea is we have a sample that we want to image, and instead of using just a normal wide field system, we want to use something that looks with a light sheet from the side, but only uses a single objective. And that you do with an oblique plane microscope. So basically, the light sheet comes from the same objective as the one that you're using for collecting the light. And you can do that. It's just beam shaping. You put that into your microscope via the correct mirror. This is now a telescope. This is a telescope. So we have access to a Fourier domain. And now if we turn this Galvo mirror here, we are scanning this oblique light sheet through a sample, descan it over the same system, and then we end up in this remote space. So very importantly, objective 1 and objective 2, they form a perfect imaging relay. So the total magnification between these two is exactly the ratio of the refractive indices between whatever your sample is and generally air, whatever this thing is in. And that gives us a perfect image or a perfect volume of what we're looking at here. So now we can conjugate this image plane at whatever angle we have to the image plane of this objective 3. And for really high resolution imaging, you can buy these snouty objectives with a glass tip. And that works very nicely for high resolution imaging. And this is what we used it for. We have a working one now in Tromsø, very good PhD student who built it. And we looked at, for example, on these fish cells, which is a big thing in Norway. It's after oil and gas, it's the biggest industry, fish farming. So they're very interested in fish. And what we're looking at here is the immune system of the fish on the very outside. So if you take a fish and you pluck a little scale off of its side, you have these cells, they're called keratocytes, and they start crawling off and go all over your microscopy dish. And that's how they look like in a DIC microscope. So let's zoom a bit in. And this is the image quality that we get with our oblique plane microscope when we label them. So it is very close to 250 nanometer resolution. And if you look very closely, we can even see like these little nano channels that no one really knows what they're for because no one could see them before, coming out of these cells. And they're really generated all the time and are looking around, even between cells. So it's going to be interesting what these are actually doing when we do imaging for a longer period now of these cells. So that's a really cool system. But what if you want to look at a big sample? And we wanted to do that together with people at EMBL in Heidelberg. Well, this is the sample we want to look at. It's a Nematostella, so it's a kind of sea anemone. And you can see on the scale, this is a very big sample that's just swimming through a bright field microscope here. You can't just use a objective with a smaller NA to get a bigger field of view, because when you do that, well, also your image plane here changes, and you basically get no light through anymore into your sample. So this does not work. So how do you do oblique plane imaging for very large samples? And one idea that we had was, well, just use another objective, just not at a right angle. So you can still fit a dish in here to put your sample in. As long as everything is below the dish surface, you're good to go. And now we can have a very shallow angle, and we use the same trick to adjust our image plane to match the illumination plane. So we did that. That's how it looks like. You have one illumination objective, you have one detection objective with a lower NA and a much bigger field of view. And if you put a dish in here, you still have to check what is the image quality of our illumination beam that we get through. So after a bit of CMAXing, you can see that within hundreds of micrometers, we are still diffraction limited on the illumination beam for a respectable 0.1 NA. That's quite good for this type of imaging. And that's through a 1.5 cover slip. So if this is a thinner cover slip, then this is even larger. And if you take the dish away and just have your sea anemones swim in here, just in this water bath, then you're basically unlimited to the NA that you want to use. It's all going to be diffraction limited. But the problem is, if you go higher NA, well, your light sheet becomes shorter, but it's still a big sample. So there, what we did was we made use of this axially scanned light sheet microscopy, just in an oblique way. So the waist of this light sheet is actually now sweeping in this direction. And when we conjugate this image plane with our camera, we can use a rolling shutter to only get the in-focus portion of this light sheet. The same principle as you would get in normal ASLM. So go through, it all ends up on the camera. And if you do that and compare the results that we are getting on just a 3D sample of beads, now this is 50 micron, so this is a lot bigger. It's a massive field of view. If you take a low NA illumination and just have it static, just go through a volume, this is the set resolution that we are getting. So it's close to 6 to maybe up here a bit higher even than 7 micrometers. With the high NA, but still static illumination, we can get very close to about 3 to 4 micrometer resolution in set. That's pretty good, but it drops off very quickly. And you can see that here as well. Now, these samples, they are more than 100 micrometers. So with this axial sweeping, we actually get, and this is the yellow line here, we get it almost over 500-600 micrometers in depth to be below 4 micrometers resolution. And with, well, 2 by 2 by 4 micrometers, you're good to go to image these cells and look at individual, almost individual cells even in these living nematostellar samples. So here's now what you get. This is just one plane, a lot of autofocus light, and this is how it swims in 3D through our imaging volume. This is a comparison between if you don't have this axial scanning on and with the axial scanning, and you can really see that the axial resolution makes a jump in performance. Now, what is very cool though is if you do it right, then and you're imaging with a very fast sweeping axial scan, you're not even reducing the speed at which you can image. And it turns out that we can go at no loss in speed with the system because there's so much fluorescence we still get out that it's good enough to run it as the fastest possible frame rate of the camera. So there is no additional waiting time involved. If you want to see all the details of the system, and this is the paper, it was spearheaded by Jon Rickard and Samuel in Heidelberg, and it was together with Robert Prevedel's group. And that system is also still in Heidelberg. So if you want to use it, this one is in Germany. That's not in Tromsø. So yeah, then let me start to wrap up. So what I showed you was the Solis technique, which is an oblique light sheet imaging system that captures entire volume per camera frame, optically sectioned by basically doing line scanning confocal in multiple image planes. Variety of multifocus elements you can combine with it. And I showed you oblique plane microscopy and how to do that with axial scanning to remove out of focus light a bit more effective with their scattering, but also to increase the axial resolution over a very large sample. This is the team that I've been working with. A lot of the people in my group, a lot of collaborators. James is a good collaborator and friend in Cambridge, and people in Tromsø that we work a lot with on getting different samples. This is the paper for Solis. If you want to learn more about it, actually got an innovation award recently, and this is the QR code that will lead you to that paper. This is the paper that I just had on the mesoscopic axially scanned light sheet imaging. This is the multifocus paper, and I think now I have all the papers presented that are part of here. Let me go back to the people because they did a lot of work. So thank you very much for having me here. Thank you all for listening. I'm happy to answer any questions now.